yourself. First of all, hello everyone. I am Ajui Kim, an organizer of today's webinar, and currently I am a master degree student under supervision of Professor Kwang Yun Cho, and my research interest is tumor heterogeneity and the subclonal evolution in cancer.
Hello everyone, my name is Nam-hee Kim. I'm the moderator of this seminar. I'm studying in Professor Kwang Yun Cho's lab, and my major is systems biology. Thank you.
Hello, I'm the panelist Jung Kim. I'm in Professor Jung Kyun Cho's lab, and my research interest is cancer immunology.
Hello, I'm Sumin Lee. I'm also a panelist, and I'm in Kwang Yun Cho Professor's lab, and my research interest is cancer precision medicine.
Hello, my name is Jaemin Lee. I'm the first presenter of today's webinar. I'm currently in master degree program in the Professor Kwang Yun Cho's lab. My research interest is systems biology in cancer. Thank you.
Hello, I'm the panelist in today's seminar. My name is Seo Ka-young, and I'm studying on omics data analysis in Jung Kyun Cho's lab as PhD student.
Doyeon-an, please introduce yourself, please. 소리도 안 나.
Can you hear me? 소리 들리세요, 혹시?
네, 들려요.
네. Hello, my name is Doyeon-an.
내가 멈췄니?
I'm from bioengineering and brain department, and I'm presenter, and I will introduce single-cell sequence technology in cancer research.
Yeah. Yes, okay, we will start our seminar. Our seminar's title is sorry. Our seminar's title is Usage of Various Sequencing Data in Cancer Research. So first presenter will talk about the bulk RNA sequencing data with brief introduction. And second presenter, Doyeon-an, will introduce about the single-cell RNA sequencing data. So we will start.
Let me present how bulk RNA-seq data have been used in cancer research. First, I will introduce various NGS technologies and among them, I will discuss more detailed experiments, procedure, and analysis method of bulk RNA-seq. Also, I will explain the case of applying them to cancer research, then talk about the limitations of bulk RNA-seq research. The next generation sequencing is a method of rapidly reading vast amounts of genetic information by breaking down a single gene into countless pieces, reading each species at the same time, and combining them using computer technology. The next generation sequencing can analyze the entire field of genomes across DNA levels, RNA levels, and epigenetic levels. For this purpose, various analysis platforms such as whole genome sequencing, whole exome sequencing, target sequencing, and whole transcriptome sequencing are included. Whole exomes and whole genome sequencing are becoming popular since they can capture the gene or genome level genomic alteration. This method of analysis is useful for massive genomic information production and the discovery of new genomic variations. But there are limitations including long-time data analysis and application difficulties due to vast amounts of data production, and target sequencing has emerged to overcome them. Target sequencing covers the hundreds of genes whose alterations are well known for their roles in the cancer pathology and that are targetable by chemical or immunology agents. In cancer patients, the target sequencing has started to produce clinical relevant findings such as actionable mutations for the patient. RNA sequencing, also called whole transcriptome sequencing, can quantify the level of individual genes and also identify the structural changes in the transcriptome that also play roles in the development of cancers. Usually, gene expression is a measure of the abundance of protein. DNA goes through an RNA state before it is translated into a protein. RNA, not DNA, plays a key role in gene expression research because it is calculated that the more transcripts, the more protein are expressed. Let's find out more about RNA-seq. Since bulk RNA-seq was developed in 2008, it has become a popular genomic tool in the life science field and is shaping nearly every aspect of our understanding of genomic functions. Bulk RNA-seq takes more than 60% of all next generation sequencing projects. It is the most widely used genomic technique for studying the transcriptional landscape and altered molecular pathway in human cancer. RNA-seq consists of 4 key steps: total RNA extraction, library construction, sequencing, and data analysis. First, RNA is extracted from the cells or tissues. Second, subset of RNA molecules are isolated using a specific protocol, such as the poly-A selection protocol to enrich for polyadenylated transcripts or a ribodepletion protocol to remove ribosomal RNAs. Next, the RNA is synthesized into complementary DNA by reverse transcription, and sequencing adapters are ligated to the ends of the cDNA fragments. Following amplification by PCR, the RNA-seq library is ready for sequencing. After sequencing, this alignment to reference genome is the first step to do. Second, the aligned reads are assembled into transcripts using reference transcript annotations or de novo assembly approach. Next, the expression level of each gene is quantified by counting the number of reads that align to each exon or full-length transcript. The transcriptome sequencing data obtained through mapping and the quantification process are used in the differential expressive gene analysis. DG analysis is one of widely used bulk RNA-seq analysis to compare two or more samples or groups to find genes that significantly differ in expression. For example, in figure A and B, by comparing genes expression level of colon cancer patients with normal people, it is possible to find biomarkers which are genes that are uniquely high in colon cancer patients. Following the DG analysis, hierarchical clustering method is carried out for clustering of groups with similar expression patterns of genes, which is useful for finding and classifying eugenetic subgroups within a group. Gene set enrichment analysis can also be performed based on gene expression difference, which helps to understand the mechanism of disease development by ensuring that genes belonging to a particular ontology or biological pathway are significantly contained in differential expressed genes. How kinds of analysis method that using RNA-seq data in cancer research? I will introduce two different research cases that use bulk RNA-seq in cancer research. As you can see in the left top of the slide, the first study is that finding biomarkers related to the endocrine therapy of luminal breast cancer. Usually, patients with luminal breast cancer are prescribed for endocrine therapy despite ongoing advance in endocrine therapy today. A proportion of patients ultimately develop endocrine resistance, resulting in failure of therapy and poor prognosis. As part of the increasing concept of personalized medicine, the authors of this paper recognized the need to identify the predictive markers of endocrine therapy response at an early stage. They saved DEG by dividing it into cases that responded to the endocrine therapy and cases that did not. Also, they identified DBN1 as a key DEG associated with response to hormone treatment. Next, authors assessed large, well-characterized cohorts of primary luminal breast cancer with long-term follow-up DBN1 at the mRNA and protein levels as a prognostic marker in luminal breast cancer, as well as its potential for predicting the benefit of endocrine therapy. And difference of DBN1 protein expression between responsive and unresponsive patient samples to endocrine therapy can be observed through immunohistochemistry analysis. You can see a result in top right. As you can see in the bottom right on the slide, which show patient survival plot, DBN1 positivity was associated with aggressive clinical pathological variables and poor patient outcomes. Importantly, high DBN1 expression predicted relapse patients who were subject to endocrine treatment. Therefore, they confirm that DBN1 is an independent prognostic marker in luminal breast cancer. In the second study, as you can see on the left top slide, this research combines network analysis with bulk RNA-seq data to identify transcription factors that significantly and maximally regulate such a biomarker gene. That factor, a framework uses a computationally inferred context-specific gene regulatory network and applies topological, statistical, and optimization methods to identify regulator TFs. This study deals with asthma, but it can be applied equally in the cancer. Application of that factor to a multi-gene expression-based asthma biomarker identified ETB4 as the biomarker's most significant TF regulators. Through these TF's siRNA knockdown experiments in an airway epithelial cell line, they showed a significant decrease in the expression of cytokine associated with asthma and validated the NEC factor's top scoring findings. This is the other research that uses DG analysis from bulk RNA-seq data as well as network analysis. Besides these researches, there are various types of cancer research using bulk RNA-seq. But there are some limitations in the bulk RNA-seq research. Bulk RNA-seq requires at least an RNA in nanogram unit. However, the cells of ordinary mammals have RNA in amount of picogram unit. When we don't obtain a sufficient numbers of cells, bulk RNA-seq is impossible to perform. Also, it is important to keep in mind that bulk RNA-seq reveals only an average gene expression profile from the samples or tissues of interest. Most tumors contain heterogeneous cell populations, including malignant cells, immune cells, fibroblasts, and vascular cells. This heterogeneity exists not only in the same tumor types from multiple patients, but also within various tumors from individual patients. So this average gene expression profile from tumor tissue can potentially weaken the true signals from a specific cell type that may drive tumorigenesis or resistance to treatment. This weakness can be overcome by the alternative RNA-seq technology, single-cell RNA-seq, which the next presenter will explain. This is the end of my presentation. Thank you for listening.
Thank you. Next, second presenter will talk about single-cell RNA sequencing data and future direction.
I'm sorry, wait a second. 죄송합니다. 혹시 들리시나요?
네, 들려요.
I'm sorry. Hello, my name is Doyana. I will briefly introduce single-cell sequencing technologies in cancer research. Single-cell sequencing examines the sequence information from individual cells, providing a higher resolution of cellular differences and a better understanding of the function of an individual cell. This figure shows one of the capturing methods to take only single cell from bulk tissue. Until now, most sequencing analyses were done with bulk tissues rather than single cells. This is a bulk tissue, and using some instruments, we just grind this whole tissue to get bulk RNA input. Just because we use the whole RNA input of tissue, we can just observe average gene expression from our cells, and cellular heterogeneity is hidden. Unlike bulk analysis, single-cell analysis use a single cell as input, so we can get distinct expression profile from each cell type and find heterogeneity and subpopulation expression variability of cells. Because of these advantages, the researches using single-cell analysis quickly increases nowadays, including cancer research. Roughly, there are some fields in omics like genomics, epigenomics, transcriptomics, etc. Among these, the field where the single-cell analysis are actively used is transcriptomics. This slide is about the fundamental principle of single-cell RNA sequencing. The first step is cell isolation step. There are several methods to isolate a single cell, and these days, droplet-based method is the most widely used technique. As you can see here, a single cell and a single barcoded bead should be isolated using partitioning oil. A barcoded bead includes oligo-DT primer having cell barcode, UMI, and poly-DT sequence. The second step is reverse transcription. This poly-DT sequence of oligo-DT primer is used to capture mRNA poly A tail. Then the reverse transcriptase makes cDNA molecule, including mRNA sequence information, UMI, and cell barcode information. So we can prepare cDNA library from single cell. This green barcode are like cell identifier. Using these barcode sequences, we can know which cell this mRNA sequence is from. And each UMI sequence is like mRNA identifier. So we can differentiate lots of mRNA molecules in a single cell. Using this library, we can make a matrix. The row means each cell, and the column means each gene. This matrix can be used for further analysis. Using the expression profile of single cells, first, we can do graph clustering. As you can see, each cell type forms a distinct cluster, and using marker genes, we can annotate each cluster. Like this figure, obtained cancer tissue include not only cancer cells, but also other cell types like immune cells and even normal epithelial cells. So this clustering step can be very critical and can be used research tumor microenvironment. So in this process, we can even discover new cell type. In the terms of cancer research, we may identify evolved subpopulations in cancer. Secondly, we can do trajectory analysis. Based on gene expression profile, we can guess which cluster is origin and which cluster derived from the origin. This analysis is used in development biology field, but it can also be used to trace how cancer cells evolve in a patient. So as I said before, the most part of single cell techniques is about transcriptomics, but there are some movements to use single cell techniques in other fields. ATAC-seq is one of the sequencing methods to identify open chromatin regions, including gene promoter, actively transcribed genes, etc. This single-cell ATAC-seq make this method performed at single-cell level. I think the main difference from SC-RNA-seq is SC-ATAC-seq uses materials from nucleic extraction, not RNA extractions. And this is a paper in cancer research field using single-cell ATAC-seq technology. As you can see here, this paper compare bulk ATAC-seq with aggregate single-cell ATAC-seq results. And this shows single-cell ATAC-seq result is consistent with bulk-seq result, and single-cell ATAC-seq provide more information at the resolution of single-cell level. And finally, I will introduce the most promising company in the single-cell sequencing field, 10x Genomics. From the initiation of single-cell gene expression technique in 2016, 10x Genomics developed many tools and instruments using single-cell techniques at other fields like ATAC-seq, CMV, and etc. And 10x Genomics continuously develop other tools and instruments to use these single-cell techniques in many fields. Also, this figure shows, this company shows exceptional financial profile. I think this company and other companies and researchers in the world continuously shows good usage of single-cell sequencing. And the market and technique of single-cell sequencing rapidly increases in few years. And also, this will help rapid development in cancer research. Thank you.
Okay, this is the end of presentation part. So next we will discuss about the data analysis of single cell and bulk RNA sequencing data. First, panelists will introduce about how to improve the analysis of single cell RNA sequencing.
Can you hear me?
Okay.
Hello, I'm the first panelist in today's seminar. I'm gonna talk about the methods for improvement of single-cell RNA-seq data in this session. There are some limitations in single-cell RNA sequencing data. Unlike bulk RNA-seq data, the majority of reported expression levels in scRNA-seq are zeros. Also, due to the prohibitive costs and ethical issues, low number of biosamples are available. Therefore, there are some famous attempts to overcome these limitations. First, let's talk about the problem of a large proportion of zeros for expressed genes in scRNA-seq data. There are two reasons why the data shows zero expression. The gene was not expressing any RNA at the time the cell was experimentally isolated, and processed prior sequencing, or the gene was expressing RNA in the cell at the time of isolation, but due to limitations of current experimental protocols to detect low amount of RNA in a cell, the gene was not detected. And also, as you can see in the figure, the proportion of genes with zero expression level varies substantially across single cell and studies compared to bulk RNA-seq samples. Not only does the zero fraction strongly correlate with the first principal component, but it also affects the further analysis. To address the increased sparsity observed in scRNA-seq data, recent work has led to the development of imputation methods in a similar spirit to imputing genotype data for genotypes that are missing or not observed. The imputation methods can be categorized in several types, such as probabilistic model-based, smoothing-based, deep learning-based, and low-rank matrix-based methods. As a result of good imputation, the gene expressions are accurately recovered as similar as bulk RNA-seq level and show better performance in further analysis. The next improvement is data augmentation. I want to introduce a recent study about in silico generation of scRNA-seq data. In this study, authors used generative adversarial network to create scRNA data. GAN structure consists of generator and discriminator. As you know, the generator generates fake data and discriminator discriminates the fake data from the real input data. After training, the GAN finally generates synthetic data, but very close to the real data. Using this deep learning model, the authors generated realistic RNA-seq data of artificial cells in certain clusters, so the generated data cannot be distinguished from the original data in the dimension reduction method. And they also validated the generated data using some marker genes expression. Augmenting sparse cell population with GAN-generated cells improves downstream analysis, such as the detection of marker genes, the robustness and reliability of classifiers, the assessment of novel analysis on algorithms, and might reduce the number of animal experiments and costs in consequence. And this study can also be applied to augmentation of other biomedical data types. The contents I want to deal with in this session is over. Here's references of my talk, and thank you for listening.
Yes. Thank you. The second part is the case study of immunotherapy using cancer sequencing data.
Hello, I am the second panelist, and our presenters have covered the basics of single-cell techniques and its application. In this session, I'm going to focus on usage of single-cell technique in cancer immunology and how it is changing the field. Cancer immunology via bulk sequencing was difficult due to small cell immune population. If we take out tumor mass, the frequency of immune cells are relatively low, and thus it's difficult to determine immune environment in given tumor. However, thanks to single-cell technique, we are now able to examine immune cells specifically. In this review paper published last year, the authors listed 8 main topics enabled by single-cell technique. Briefly, it involves identifying novel immune subtypes, reconstructing the trajectory. This part was mentioned by our presenter. Studying immune process, integrating single-cell omics data, and etc. Among these topics, I'm going to introduce cancer-related topics. The first paper I'm going to introduce is about understanding regional immunity in tumors. As you probably know, different tumor types exhibit different microenvironment. In this paper, the researchers made landscape paper of infiltrating T cells in liver cancer. They performed single-cell sequencing on immune cells and were able to distinguish different types of T cells as shown in this figure. Their main focus was characterizing exhausted T cells, which are essentially all T cells that are unable to exert cytotoxic activity. Through comparison between the effector T cells and exhausted T cells, they were able to find differently regulated gene called LAYN. If LAYN is expressed, the T cells will lose ability to make interferon gamma, which is a cytotoxic agent, and Treg cells will also turn into suppressive T regulatory T cells. What this signifies is that they were able to find T cell developmental trajectory that is related to immune evasion. If we are able to reverse development into exhausted T cells, the cytotoxic activity will continue, which means that the person is better able to fight cancer with his or her own immune system. The next topic I want to introduce is single-cell T-cell receptor repertoire sequencing. T-cell receptors, as well as B-cell receptors, are known for its diversity. They are very heterogeneous because different T-cell receptors recognize different antigens or antibodies. They accomplish this diversity through recombination, which is called VDJ recombination. They parse and add fractions from V, D, and J sequences to make a receptor, but in bulk sequencing, it is impossible to account for all T-cell repertoire using the sequencing method. Instead, we were only able to sequence TCR of a major T-cell clone. However, in single-cell sequencing, we are now able to sequence T-cell receptors of individual T cells. Using this new technique, this research group made organoid treatment model and compared it with fresh tumor. So they took out tumor samples and they grew organoid from these cells and treated it with immunotherapy. And then they compared these cells from the fresh tumor cells. As you can see here, this is the fresh tumor and this is the organoid. You can see that the blue and the green spots, which are T helper cells and T cytotoxic cells, their population grew quite significantly. And then they sequenced the T cell repertoire from the same cell. So they did single-cell RNA-seq and single-cell VGJ-seq in the same cell. And as you can see here, compared to the fresh tumor population, many T cells in the organoid population went through TCR chain rearrangement. This means that the TCR combined the VGJ sequence so that it can better recognize the tumor cells. Through this research, they were able to prove role of antigen-specific T infiltrating lymphocytes, eliciting tumor cytotoxicity. Finally, they were able to examine changes in TCR sequence and immune cell expression upon immunotherapy treatment. So in this session, I have introduced two studies. And like these two studies, single-cell techniques enables new types of researches that bulk sequencing were not able to accomplish. Due to its high specificity, we are now able to account for immune cell activity and immune cell activity only. I believe that the next breakthrough in sequencing technique will be long read sequencing in single-cell manner, and I am eager to see how development in sequencing technology will enhance and open new chapters in cancer immunology research. This is the end of my presentation, and thank you for listening.
Thank you. Last part is whole genome sequencing in cancer research.
안 들려요.
Sorry, I will restart. Hello, I'm the last panelist, Sumi Lee, and I'm gonna discuss about applications of whole genome sequencing in cancer. And whole genome sequencing can be applied at both bulk and single cell level, and it's an application of next generation sequencing. And this is used to analyze the entire genomes at once, and as a result, we can identify genetic variants. And using these genetic variants, we can apply it to inherited disorders and mutations for cancer. And the basic flow is that we extract DNA from an organism's tissue, then prepare a library, and then we determine the sequence of the DNA using a machine. And lastly, we can interpret the sequencing result using bioinformatics. This has advantages and disadvantages. First, it produces a large data in a short amount of time, and the data is comprehensive. And we can also identify unknown genes that contribute to diseases. However, since it analyzed the entire genomes, there is abundance of information. So to overcome this issue, you can rather use targeted sequencing or whole exome sequencing. Next, we will look at the application of whole genome sequencing in cancer genomics and precision medicine. So somatic protein-altered mutations occur in most cancer types. Therefore, it's important to identify those mutations. By using whole genome sequencing, we can identify the mutations in non-coding regions, coding regions, and structural variants. Therefore, it produces abundant mutation information. And this table represents the comparison of all the sequencing methods. And you can see that whole genome sequencing covers almost all mutations. Therefore, all types of genomic alterations can be identified. Next, we will look at its application in colorectal cancer. So by integrating the next generation sequencing system, intratumor heterogeneity could be identified. This enabled drug response prediction and individualized therapy. And selection of drug combination was also possible. Therefore, clinical precision oncology for colorectal cancer research was advanced. So whole genome sequencing has a positive feature, and I summarize it into 3 points. And first, precision oncology research became possible because all these abundant mutation information could be identified. And also, tumor heterogeneity problem could be partially solved with the help of whole genome sequencing. And also, personalized cancer care became possible due to the better understanding of disease process. And many researchers suggest that multi-omics has another positive feature, and that is the integration of epigenome, transcriptome, and genome. And here's the references, and thank you for your attention.
Okay, Actually, this is the end of our seminar, but if you have any questions, please use Q&A tab. So there are no questions. So we will, this is the end of our seminar. Thank you for listening.